Good afternoon, Oxford and Europe, and good morning to Americas. Frank, who kindly joined us so early in the morning. Welcome to the first seminar of Department of Plant Sciences. And I would like to introduce Frank. And also in the beginning, maybe mention that this is kind of a blessing in disguise, having him digitally, because we have people from all around the world, from India, from Australia. So it's a great pleasure to be hosting this online. Professor Frank Dayan was born in Lyon and grew up in a little town in southern France. And he developed his appreciation for plant sciences and agriculture when he attended in high school the Institut Agricole de Fontenay, where he learned to appreciate plants. He received his PhD in plant physiology from Auburn University in Alabama in 1995 and worked as a plant research physiologist with USDA ARS, Natural Products Utilization Research Unit, for 20 years. He is now professor in the Department of Agriculture Biology at Colorado State University, and his work covers the mechanisms of action of natural and synthetic herbicides and the mechanism of herbicide resistance in plants, as well as chemical ecological studies of plant-plant interactions, so-called allelopathy. He has served as a treasurer of the Phytochemical Society of North America from 2006 and 2011, and president in 2014 until 2015. He also served as a treasurer for the International Weed Science Society from 2012 to 2016. Frank is an associate editor for Weed Science and Pesticide Biochemistry and Physiology, as well as North American editor for Outlooks on Pest Management. Professor Dayan has published 160 scientific articles and 65 reviews and book chapters. His work has been cited at least 12,000 times, and his Google H index is 60. So we very much look forward to his seminar. And now I am very pleased to give the word to Frank. Thanks a lot for joining us.
Thank you, Radka. And so I'm going to share my screen now, right here. Okay, so let me see. All right. So thank you so much for giving me this opportunity. And I want to welcome all my friends from North and South America and Europe, even my family, I think, may be watching this. So I'm very excited for them to see me in action in my career. They've never had that opportunity. And also my friends outside of more East, the Middle East and the Far East and Australia. It's a pleasure to do this presentation, which will be very different from most plant science presentations. And you'll notice the background is black, which is not customary. Normally I have a nice white background, but because of the nature of this talk, it's a more dramatic effect, and hopefully you will appreciate this as we go through the presentation. So again, the talk will be about a molecule called protoporphyrin, and also the enzyme responsible for its biosynthesis. And it's a very important macrocyclic molecule, and it's one ring that rules them all. And because I'm giving this seminar through Oxford University, I thought that would be a little nod at Tolkien, who's a professor at that university that wrote, of course, the Lord of the Ring. And in the Fellowship of the Ring, he talks about that one ring that rules them all. One ring to find them, one ring to bring them all, and in the darkness binds them. So hence the dark background. So there'll be 4 different topics I will talk about, and it's all unified across these macrocyclic rings. So we'll talk about medical issues, we'll talk about history, we'll talk about mythology, and we'll talk about plant science. And all of this will come together because of this common theme of the ring. And that ring is really synthesized by an enzyme called protoporphyrin oxidase. And it's really hard to say. So if you hear me say PPO, that'll be the abbreviation for this enzyme that's moving on your screen here. And This talk has been updated, but initially was actually published in 2011 in a paper that I co-authored with my oldest daughter when she was a senior in college. And so if you're interested, you can send me an email and I can send you a PDF for that document, which is Porphyrins, One Ring in the Colors of Life. And why is that important? That enzyme uses a substrate called protoporphyrinogen, as is on your screen here, protoporphyrinogen, and I'll call that protogen. And it's a colorless macrocyclic molecule. And the enzyme introduces 4 double bonds in these methylene bridge. And what happens is this becomes fully conjugated. And as that happens, the ring becomes bright red. It's a fluorescent red molecule. And actually, that is what makes your red blood cells red, is because you have heme, and heme is derived from this molecule, protoporphyrin. So the whole theme across the 4 topics will be that enzyme and that ring. So let's start with this ring. Why is this ring so important, the protoporphyrin ring? Well, it's because it can be chelated with different metal dications. So I'm going to give you some examples of maybe molecules that you may be familiar with that are derived from this macrocyclic backbone. Well, if you add an iron, if you chelate an iron at the center of this molecule, that leads to heme. And all of us are familiar with heme and its red color. What's interesting is if you put a magnesium inside this ring, and there's further modification, the ring actually turns green and leads to chlorophyll. But there's other important colorful molecules coming out of this. If a cobalt is chelated into this ring, you end up with vitamin B12. And if you chelate a nickel, you end up with coenzyme F430, which is an important cofactor for some early bacteria. So it's across life. The entire life on Earth depends on this molecule. So let's focus on the biosynthesis of protoporphyrin and heme in human. That is a very short representation of the biosynthesis that starts with glycine and succinyl-CoA and leads to protoporphyrin and heme. All of these are colorless, and of course this one becomes red, and then the last product is also red. For those of you who don't specialize in biochemistry, what you have to realize is that there are enzymes responsible for the biosynthesis of every one of those steps. And these are the names of the enzyme on the left, each one catalyzing one step to the other. And as far as humans, what's really interesting is there are genetic diseases where there are deficiency in these enzymes for every steps. So, and this leads to a set of disease called porphyrias. So that there are 1, 2, 3, 4, 5, 6, 7 different types of porphyrias, depending on which enzyme is deficient. And depending which one it is, it can have a different level of medical implications. Again, so I'm not a medical doctor, so I'm just going to really go quickly over this because it leads to the important historical facts. So the one that I'll be focusing on is this enzyme right here, variegate porphyria. That is a deficiency of the enzyme PPO that converts that colorless precursor into the photodynamic protoporphyrin. And so when there is someone inherits this disease, there's different levels of medical consequences. Oh, I guess before I do this, I want to tell you a little bit about the enzyme. So this enzyme, the PPO enzyme, I'm just showing you basically the catalytic domain of that enzyme. And there's some important features I want you to know because this will come back later on when we talk about herbicides. And all PPOs have these 3 features. There is a glycine at the bottom of the pocket here. This is actually on top of a helix, and there's a glycine that sits on top. There is a FAD, which is a cofactor that's necessary for the enzyme to do the oxidation, that sits kind of on top and forms the roof of the catalytic domain. And all of these enzymes have a very important arginine that is positioned on the left side of the pocket in the way I'm orienting it here. And this arginine is involved in stabilizing the enzyme. So the next slide would be exactly the same one, other than the substrate being in the pocket. And to make the point that I was saying, the substrate, the porphyrin ring right here sits on top of the glycine, almost like a crown, and it's positioned exactly in the right distance for hydrogen bonding between the arginine and the propionate group. So this carboxylic group from one of the side chains of the porphyrin is interacting with this arginine, and it positions it in the right place so that it's at the correct distance for the FAD to have what's called a hydride abstraction. It can remove a proton from the from PPO, and that leads to the oxidation reaction to lead to the formation of protoporphyrin 9. So this will come back. So I just wanted to introduce it now because this will be important later on. So let's talk about the disease real briefly. This, as I said, there's 7 different types of porphyria. This is a little description. When the pathway is disrupted because of those mutations, especially that one that affects PPO, porphyrin, instead of being localized where it needs to be, like normally it's in your blood cells, in your bone marrow, when you have the disease, the porphyrin comes out of that natural environment and accumulates in your skin. It can accumulate in the teeth. And then when it gets in your skin, I mentioned protoporphyrin is red, it's photodynamic. It actually can react like chlorophyll in the sunlight, but because it's in your skin, it causes lesions. So that people that have porphyria, some forms of porphyria, have symptoms like this. They have lesions in the skin. They can have blisters when they go in the sunlight, which is a very debilitating disease. All right, so that was all I wanted to say about the medical condition, because this is a good segue to the historical part. You know, why bring history into a scientific talk? I need to close my door because people are coming in. Sorry about that. All right, so there's a movie called The Madness of King George, which is a really good movie. And actually in the movie, they actually give an indication that King George III, who was king during the American Revolution, actually had symptoms of porphyria. And on the left here, you have a quote from one of the doctors of the king, and that's in the medical record of King George. It says that the water, meaning the urine, was of a deeper color and leaves a pale blue ring upon the glass near the upper surface. So to be consistent with the theme of the ring, I thought this was a great description. And that blue color is actually a symptoms of porphyria, because when porphyrin accumulates outside of its natural environment, our body try to get rid of it, and as it's being metabolized in the body, it actually turns blue or purple. And so it's a very telltale sign of somebody having porphyria when the urine, instead of being yellow, has a bluish or purplish color. And that is a good indication that King George had that disease. It's been medically documented. And so when bad cases of porphyria can affect the nervous system and cause mental illness. And this is something I learned as I was studying this topic, is that actually since King George suffered or was thought to suffer from porphyria, scientists in modern time, scientists have looked back in those medical records, and they found all the telltale signs that he had porphyria. Some typical symptoms will be abdominal pain. Of course, the color of the urine that I mentioned, that's not yellow, but more of a purplish or blue, and sometimes leads to paralysis of the arms and legs. And in many of the medical records of King George, there are mentions of these symptoms. And in fact, and so I'm citing the paper where I found this information, because of course I'm not a medical doctor and I wanted to support my statements here. And there are actually records of King George having a special area of Windsor Castle where he was basically closed in toward the end of his life, because that disease really had affected his mental capacity. Okay, so why is that important? It's because, of course, he was king during the American Revolution. And some historians have hypothesized that if King George had not had porphyria, which had affected his ability to maybe reason at times. And it's I should mention that there are instances, so you can have the disease and have no symptoms, or you can have the disease and sometimes you have a wave of a crisis and then it gets better. And there's several crises. And apparently in King George's life, there were 7 documented instances where the disease took over. And so some historians have postulated that possibly had King George been maybe more clear-minded, might have been able to see what was going on in the colonies of the US, and their reasonable demand of, taxation without representation was not acceptable, and they wanted to have some kind of representation, and that was never permitted, and that led to the American Revolution. So here you have an instance of a single point mutation on one enzyme that affected one person that may actually have changed the course of history, which is really interesting to consider, that a single amino acid substitution could have had such a dramatic impact on history. So this is to support the importance of that enzyme PPO. And we talked about the consequences from a medical perspective. And this is just one example of maybe a historical consequence of this mutation, because it affected a leader of the leader of the world at the time, King George. So I mentioned that this disease was genetically inherited. And these scientists in the late 60s actually looked at the medical records of all the ancestors of King George III. And of course, they have no proof that these people have porphyria, but they made a note if in the medical records there were examples of symptoms consistent with porphyria. And so actually it's pretty well known that Mary, Queen of Scots, which is the mother of the first King of England, was quite a woman and had quite a reputation. And some of it was because she had some, what's the word? I don't want to say something that's not correct. But she may have had some symptoms that she may have had porphyria. And if you look at the family tree, King James I also had some potential symptoms of the disease. And if you look down the family tree, again, so I have redrawn these from this paper that I'm listing on the top right. Basically, King George III and one of his sisters had symptoms. And then all of his offsprings also had symptoms of the disease. So, there's some credence to the fact that maybe it actually was introduced in the family from way back. And sometimes you can have, I mentioned, you can have the trait without the symptoms. And so there's some family members that did not have any evidence of the disease. Now, what's interesting is Sophia, which was free of symptoms, was married into the family of Prussia. And all of a sudden, we start seeing symptoms of the disease on that side of the family. And as it was not uncommon in Europe at the time, families remarried into each other for alliances, and so I'm not familiar with the history between Charles and Augusta. But what's interesting is they had no symptoms, but now Caroline, their offspring, had the symptoms. And then to top it all off, their daughter Caroline and George IV married, and they had a princess, only one child, that died as a young woman. So I think there's some pretty good evidence that maybe that is really historical, that porphyria was a trait that was passed down that family tree. Now, I mentioned that you can have the trait without having the symptoms, and it's known that some environmental conditions that can trigger instances of the disease. So you may have the trait and you may be more sensitive and something happens. And so this 2005 paper is really interesting because we know that some metal can cause, exposure to some metals can cause porphyria to express itself. And so this group in 2005 analyzed the composition of the hair of King George. So they had actually a lock of hair in a museum at the British Science Museum. They took 2 strands of hair. They analyzed the composition, the metal composition in the hair, and compared it to people of that time. And so this is really interesting. So they used IPC mass spectrometry for this. And they looked at the amount of arsenic in the hair of regular people, which is about, I don't know, 30, the unit, it's a relative measurement, but it's about 30 maybe on average. And this is the amount that was present in the King George's hair. It's, twofold magnitude increase in the amount of arsenic in the hair. And they did some back calculations based on known exposure, and it is estimated that King George received between 1 and 2.5 milligrams of arsenic per day. And of course, that would have been a trigger for the porphyria. There are other clusters are known to exist, let's say in shipyards. Some of the paints that are used on boat hulls can maybe lead to porphyria as well. So there's a lot of unknown about why you can have the trait and not the evidence of it, and that there are environmental triggers that lead to this. And I think there's still more research going on into this area. So now, how does this affect mythology? So we talked about the medical implications. We talked about the historical implication. Now, how about mythology? And this is really interesting. I found this little paragraph from Bram Stoker's Dracula. And the main thing is when Mina, which is Dracula's love interest, she's also the doctor's love interest. She gets beat and he's afraid something's going to happen to her. So he sits her in a place and draws a circle using wafers, church communion wafers, and makes a ring around her to protect her against evil spirit. So because of the theme of the ring, I thought that would be an interesting little quote to mention here. But let's get to it. Vampires. What do we know about vampires? Well, it's a legend, obviously, there's no vampires. But in the legend, they are from Transylvania. They tend to be pale. They are sensitive to light. And they have fang-like teeth, right? Typical symptoms. And I have this picture on the left, which is another movie. I don't know if I would recommend it as much as The Madness of King George. This movie is actually Interview with a Vampire. And these two guys right here, that's Brad Pitt, and this is Tom Cruise. About to bite Brad Pitt. Okay, so this is the legend, right? And so how has this legend come to be? And as is not unusual for mythology, there's very often true historical facts in the background that have been used to elaborate a very crazy story, right? Such as the vampires. Well, it turns out that Transylvania is a real place, right? It's a wooded area in the Hungarian Romania area of the world, of Europe. And it's actually the birthplace of Count Dracula. And Count Dracula was actually a true historical person. And I'll mention that. His name was Vlad III. He was a prince of that area called Wallachia at the time. And he was a vassal of the king of Hungary. And that's actually the castle that can be visited, right? Of course, he was not a vampire. He just happens to be I guess what people would consider a soldier that actually was born in 1431. And his nickname, Vlad III, was Vlad Tepes. And Tepes means the impaler. And that's because he liked to impale his enemies. And this is the painting of Vlad Tepes. This is an interesting wood carving on the bottom left, where you can see him having a feast of a meal, surrounded by people being impaled and people being dismembered. So basically, Vlad was actually very cruel, and he fought off the Ottoman Empire that was invading Europe through the Alps, going through Hungary and Romania, and to scare his enemies. So he was not a vampire, but as a scare tactic, he would drink human blood. And he would just have meals around people being impaled and beheaded. It is believed that he's killed more than 130,000 Muslims. So a very ruthless person, a very cruel person. And what is also a true fact is that his father's name was Vlad Dracul. And the Dracul was actually a military unit with the symbol of a dragon. And Dracula just means son of Dracul. So Vlad Tepes was Dracula, the son of Dracul. And he was a very cruel person that, did horrible things to his enemies and drank their blood and stuff like this. But he was not a vampire. Now, what you have to imagine is, of course, there was no widespread communication, but people did travel. Merchants traveled from Europe all the way to Asia, having to go through the silk trade. And so some people did travel through this area and would encounter this region of the world where Dracula was there killing people and doing very cruel things to them. And so that may have been one part of the story of Dracula, the name and the cruelty of it. Now, what's also interesting is that in those remote parts of Europe, and this happened all over Europe, some of those remote valleys had very, very little traffic. And people tend to marry, into the population that was around them. And actually, scientists have found that there was a small population in Transylvania in those remote areas that had a higher incidence of porphyria. And that's because of the inbreeding within those populations. Instead of being a rare trait, it was a more common trait. And so now you have these populations in Transylvania, which is the same area of Europe where Dracula was. And this population had porphyria. And so what happens is when porphyrin gets into your skin, as opposed to your blood and your bones, then your skin burns when you go in the sun. Actually, porphyrin can accumulate in the back of your retina, so that when you go in the sun, your eyes burn. So these people actually avoided sunlight. They would get sunburn when they would go in the sun, and their eyes would hurt when they would go in the sun. And as I mentioned, sometimes people that have really bad cases of porphyria, the porphyrin can actually accumulate in your teeth. And so the teeth are not red, but they're more like pinkish. And it's not that they had fangs, but bad cases of porphyria causes the gum to recede. So it exposes more of your teeth. So it looks you have bigger teeth, right? So you have to imagine that these traveling merchants going through Europe back and forth would hear about Dracula being a ruthless person, killing and drinking their blood and impaling and doing all kinds of horrible things. And at the same time, in a region where people are sensitive to the sunlight and they stay indoors because when they go in the sun, their eyes hurt and their skin burn. And you can imagine these people traveling, telling the story to one another, and little by little through time, and as the story is being told over and over, the two historical facts merge to become the myth of vampires, people that would drink blood, that would be very cruel, and also very sensitive to light. Now, to make my case, I'm just going to show you a picture of one person that I found online that actually had a very bad case of porphyria to illustrate the point. So this is really what can happen to somebody with a really bad case of porphyria. Now, I want to make it clear that vampires are only a legend. A person that suffers from porphyria, such as this patient right here, is not in need of Buffy the Vampire Slayer. What they are in need of is a doctor, right? And I always want to make this disclaimer because I never know when I give a presentation like this, if I'm encountering somebody that has porphyria or somebody that knows somebody or has a family member that has porphyria. And I don't want the message to be people with porphyria are vampires. It is not. It's a disease. It's a dreadful disease, and it can be very, very harmful when in fact most people live normal lives, but it can be very bad. And it's not, people like that are not vampires. They just have a medical condition that needs treatment. So now we've looked at the importance of PPO, the enzyme, for medical conditions, how that may have affected history, and how this may have led somehow to the formation of the myth of vampires. And so, perfect, I'm halfway through the talk. So we talked about King George, we talked about vampires. How does that relate to herbicides, okay? Which is really the last 15 minutes of my talk. would be really about the plant science aspect of this talk. But really, it's interesting how biology can really cross over sciences: medicine, plant science, history, mythology, right? So let's move to the area that I am more familiar with, which is the importance of that enzyme in a plant. All right, so as I mentioned, this enzyme converts a colorless precursor called protogen to a bright red pigment called protoporphyrin or proto. And in plants, as opposed to human, in human, protoporphyrin goes into heme biosynthesis primarily. But in plants, actually, they have two pathways. You can add a magnesium into this ring, and that leads to chlorophyll, or you can add an iron into this ring, and that leads to heme. And what's interesting is in plants, you have actually two enzymes that do exactly the same reaction, PPO1 and PPO2. So these enzymes catalyze exactly the same reaction, but they have been conserved through evolution for some reason. And they have very little identity. I think they have like 30% identity at the amino acid level. So they're very different enzyme as far as the amino acid composition, but they have exactly the same activity. And in fact, the crystal structure of the plant enzyme and the crystal structure of the animal enzyme, even though they only have 30% identity in amino acids, they can be put on top of each other and they are almost identical. So the shape of the enzyme is the same and catalyze the same reaction. And they are both nuclear encoded. And what happens in plants is that the gene encoding for PPO1 produces the enzyme PPO1 that goes into the chloroplast. And there's an enzyme called PPO2 that's encoded by a gene that's also in the nucleus. And that gene product actually moves to the mitochondria. And then in the chloroplast, you put a magnesium in it, and that goes to chlorophyll biosynthesis. In the mitochondria, you add an iron into the ring, and then it goes to heme biosynthesis. So there's a little bit of background about plants. Okay, so now I've showed you the biosynthesis of porphyrins and heme in humans and animals. This is the pathway in the chloroplast, which starts with different precursor. Actually, it starts with glutamic acid. And the pathway will go like this. You have to follow a circle here. I had to fit it all in there, okay? Now, as I mentioned, of course, protoporphyrinogen, and protoporphyrin is a very photodynamic molecule. So the pathway is very regulated. Plants have a very tight control on the biosynthesis of chlorophyll. And so in the dark, there is no chlorophyll biosynthesis in the dark. And that's because in the dark, this substrate accumulates and feedback inhibits the very first step of the reaction. So it's a feedback inhibitor. And also the very first product of the reaction, the first step, if this accumulates, it also feedback. So there's 2 points of regulation at the very beginning of the pathway and at the very end of the pathway to make sure that there's no deregulation of the pathway. Now, when the sun comes up, this conversion of protochlorophyllite to chlorophyllite is actually a light-dependent reaction. It's very few reactions like this in plants, but this reaction is driven by light. And when that happens, then this feedback regulation is removed and the pathway is on, and then chlorophyll biosynthesis happens. So this is why when you grow plants in the dark from germination on, that etiolated, they're yellow because they don't have any chlorophyll. But if you put them in light, they turn green because this pathway is opened up. Now, of course, the enzyme I'm interested in is PPO right here. And that's an important one because it's a target for many, many herbicides. Actually, there was I heard a talk from BASF one time that they had done a patent search that there were more than 100,000 molecules known to target this enzyme. Of course, not all of them are herbicides, but it's a very sensitive target to herbicides. And I don't want to spend too much time on this because the way this was discovered took 20 years. Because when you inhibit this enzyme, actually the product of the reaction accumulates. And that baffled scientists forever. Because you would think if you inhibit this enzyme, well, this would accumulate, but this would go down. But in fact, they had known that this would accumulate. So scientists looked at the enzyme target site past protoporphyrin, but it turns out that it actually accumulates upon inhibition of its own reaction. So how does that happen? I want to show you an example of that. My previous boss, Steve Duke at USDA, published this paper in 1990, where that's actually right on the cuff of when they discovered how these herbicides worked. The discovery of the mode of action was done by a French group led by Michel Matrange that discovered the target site in 1989, I believe. But anyway, what you have to imagine here, this is a cell, and that's another cell. It's kind of faint because the pathway is very regulated. So these are etiolated plants, and you only have a small amount of porphyrin in the plastids, in those little red dots right here. And what happens though is when you expose those cells to a PPO inhibitor, this is what happens. You can see a great massive accumulation of protoporphyrin outside of the plastids. So the theory is that the substrate leaks out of the plastid. It is converted to protoporphyrin in the cytoplasm, and protoporphyrin is not supposed to be in the cytoplasm. That reacts with light, and that causes photodynamic damage, and it kills the plants. And that's how those herbicides work. And so again, you know, PPO has medical implications, has had historical, has had mythical implications, and now it's actually has implications in plant science because PPO herbicides are an important group of chemicals to control plants. All right, so as these herbicides are used, it's common for most other herbicides, sooner or later resistance will evolve. And this was reported in 2006. Actually, it was reported before that, but this paper that I'm listing here, the Patzol 2006, is actually when they discovered what happened to these plants. The front plants are the wild type sensitive, and you can see that they're killed with 4.2 grams per hectare of the herbicide. And the one in the back, you know, they are dinged up a little bit, but they still grow, and these will produce seeds at 100 times the rate, 420 grams per hectare. So they wanted to know how these plants became resistant. And this was published in 2006 by this group from University of Illinois, led by Pat Tranel. And what they discovered is that not only you would expect maybe an amino acid substitution, which is very common, but this was the first time in the case for herbicides that it was not an amino acid substitution, but it was a deletion, a whole codon. So 3 nucleotides actually were removed. So that the GGT was removed, and that GGT codes for a glycine. And the resistant plant had a glycine deletion. And they were able to explain how that happened. And that has to do with the genetic environment around the that glycine codon. So I'm highlighting in yellow here. You have what's called, you have what's called a macrosatellite repeat. So in this case, you have GTG, GTG. And sometimes during the replication, you can have a misstep, and it actually skips one of the GTG, and that would lead to a new strand of DNA, or that would actually miss a glycine. But what's interesting is that if you move it to 1 nucleotide or 2 to the right, then you have a TGG, TGG. So you have this microsatellite environment kind of on top of each other, and it's increased the probability of slippage and having either a GTG or a TGG deletion. So I'm sorry for those of you who are not familiar with molecular biology, but this is the name of the nucleotide that I'm talking about here, the initials. And what's interesting is whether you have a TG deletion in that sequence or a TGG deletion, both cases lead to a loss of a glycine in the way the 2 strands reanneal, right? And so that leads to a glycine deletion. Now, glycine is the smallest, most flexible amino acid in proteins. And so really the question was, when we know there's a glycine deletion, but why would a glycine deletion, which is almost insignificant in many cases, because glycine is so small and so flexible, you would think that there would be very little effect on the protein. And so I want to show you there was a glycine deletion here, but what they also found is they found the deletion not on the chloroplast gene, but on the gene that's in the nucleus that goes to the mitochondria. So that was really surprising. And what's interesting is they found that there was actually a 90 base pair extension that provided dual targeting, so that now the new protein had a deletion and then did it both in the mitochondria and the chloroplast. So real quickly, what they've discovered is that the gene encoding for the chloroplast protein would go make the protein and go to the chloroplast. The gene that goes with a deletion would go to the mitochondria. And then and you put the herbicide, you would kill or inhibit the enzyme in the mitochondria, but because of the dual targeting, now the gene that also supposed to go to the mitochondria ends up in the chloroplast. And now while the chloroplast enzyme is inhibited, the gene that's dual targeted provides tolerance because it's resistance, because this enzyme is no longer inhibited by the herbicide. So that's really kind of an interesting set of circumstances. All right, so we've been, so then at that point, I helped the group from Illinois, and that's when I came into the project. And we wanted to understand what was the meaning of the glycine deletion. So we did some biochemistry. We cloned the genes, expressed them in E. coli, and then did the enzyme assays. And we tested a variety of diphenyl ether PPO inhibitors. And first, dose response curves. The green ones are the dose response on the sensitive enzyme, and the red ones are the dose response on the resistant enzyme. And you can see there's a huge shift to the right, maybe twofold magnitude, 100 times or more resistance. So that would explain, okay, the herbicide no longer efficiently inhibits the enzyme until you put very, very high doses. And at the biochemical level, we did some kinetics. We got a graph like this. So this is the enzyme activity as you increase the substrate amount without the inhibitor, and that's with the inhibitor. And for those of you who are familiar with biochemistry, when the lines cross on the y-axis, that means that the herbicide competes for the binding of the substrate. And this was known, that was published in 1989 by the French group, that PPO inhibitors are competitive inhibitors with the substrate. And so that was known. But what's interesting is the pattern with the enzyme that was resistant was very different. That's what it looked like. You can see that the lines no longer cross on the y-axis, which changed the way the herbicide interacts with the enzyme. Okay, and as a summary, I'm going to show you. Oh yeah, so why would a glycine deletion have such a big impact on the protein? And this has to do with where the glycine deletion was located. So this is a helix number 8 in PPO, and that's the glycine that was deleted right here, this glycine. And what's important about this is that this glycine is involved in what's called helix capping. Whenever you go out of a helix into a loop, that is you have to stabilize that transfer, that conversion from a helix to a loop. And the glycine is involved in these interactions that stabilizes that. And what happened is the deletion happened there, and that completely destabilized the top loop. And this paper, and I'm sorry, I'm not citing it, but there's a paper that shows that the propensity of C1, that's C1, to be a glycine. You can see that there's threefold more chances for this amino acid to be a glycine than any other amino acids. And so that's an example of glycine playing a very important role in stabilizing the helix capping. And so we did some molecular dynamic simulations of what happened when you delete the glycine. And so this is how likely are these amino acids to be involved in a helix. And what you can see is in the wild type, it's consistently at least 90% likely to be part of a helix. However, when you do the deletion, you completely lose that last top loop of the helix is actually no longer in its preferred conformation. And this led to this helix right here being lost. With the glycine deletion, you lose a whole loop, a whole turn of the helix, and that opened up the pocket of the enzyme. So in the end, what it did is we measured the distance of the size of the pocket from the FAD to the bottom of the pocket. And what happened is that we went from a distance of 7 to 7.5 angstrom. Now, you would think that is such a small change, half an angstrom difference in the size of the pocket. Now, what you have to remember is this enzyme requires the transfer of electrons from protogen to FAD. And so by increasing that distance, now the enzyme no longer works really well. So I'm going to show you this. So back to the one of the first few slides, right? The catalytic domain, there's a glycine at the bottom, there's the FAD on top that catalyzes the reaction. There's an arginine that stabilizes the protoporphyrin ring in the catalytic domain. And the distance between that N5 and the C20 is very important because this is where the oxidation happens. And so in the resistant one, in our data, the Vmax decreased, which means that the enzyme did not work as fast, which is consistent with the size of the pocket being bigger, because now the electrons have to travel half an angstrom longer, which again seems like it's just such a small difference. But as far as electron, it's like going from here to the moon, probably. I have not made the comparison, but it's a huge change in difference. So our kinetic data said the Vmax decreased, and that's consistent with what our computer modeling illustrated, that the pocket was bigger and the distance between the FAD and the ring changed. The Km did not change, and that's because the Km is mostly driven by the shape of the pocket and that interaction between the carboxyl group and the arginine. And so again, our computer modeling was supportive of the biochemical data that we obtained. We know is that no longer the herbicides were no longer competitive inhibitors. They were more like mixed type inhibition, meaning that you could have at sometimes the herbicide and the substrate at the same time, which is different from the normal situation. And the last thing is the Ki increase, which is the inhibitory constants. Of course, that makes the enzyme more resistant to the herbicide. And now, this is my last slide. Hopefully, time-wise, I'm good. Yeah, 10 minutes ahead. So we'll have time for questions. This is an interesting case, I think, of where one single enzyme plays an important role in all of life for the synthesis of a variety of macrocyclic tetrapyrroles. And this has had an impact not only for medical everyday life things, but has had an impact in the shape of history, has led to the evolution of the myth of vampires, and has had a huge impact on agriculture. So this is the end of my talk. Thank you so much for your attention. I really appreciate it. And thank you for University of Oxford for giving me the opportunity to do this seminar for your plant science seminar series. And at this point, I will stop, and I'd like to maybe have the opportunity to answer some questions.
Thank you so much, Frank. And if our participants have questions, it would be nice if you could indicate in the comments by